Good morning, students. In this class, I am going to discuss with you about Rhizopus. Classification of Rhizopus. It comes under the class Zygomycetes, order Mucorales, family Mucoraceae, example Rhizopus. Habitat. Rhizopus is a saprophyte. It is commonly called as bread mold. It frequently grows on moist bread, decaying vegetables, fruits, pickles, jam, jelly, and butter, etc. Rhizopus classification is class Zygomycetes, order Mucorales, family Mucoraceae, example Rhizopus, and the occurrence of Rhizopus is usually saprophytic in nature, that is, it present on dead and decaying organic matter. It is commonly called as the bread mold because the fungi usually grows on bread surface, hence it is called as bread mold. It also grows on the decaying vegetables, fruits, pickles, jam, jelly and butter, etc. This is the structure of the Rhizopus hyphae. The young mycelia is cottony white in appearance. It consists of aseptate cenocytic in nature. Aseptate means septa is absent. Cenocytic is the condition in which multinucleus is present. The hyphal wall is made up of chitin. The cytoplasm shows oil globules, glycogen, vacuole and nuclei in it. This is the hyphae of Rhizopus. Rhizopus mycelia is identified by its color, that is, it is cottony white in color. These are aseptate in nature, cenocytic in condition. The hyphae wall is made up of chitin and the cytoplasm comprises of oil globules, glycogen, vacuole and nuclei in it. Let us move towards the mycelium parts. It comprises of three parts, that is stolon, rhizoidal hyphae, sporangiophore. A Rhizopus hyphae is comprises of three parts, that is stolon, rhizoidal hyphae, sporangiophore. This is the stolon. Stolon is the horizontal hyphae which creeps on the surface of the substratum. The growth of the stolon is very fast. It soon covers the entire substratum. Rhizoidal hyphae, these are highly branched finger-like hyphae produced at regular intervals on the lower surface of the stolon. These hyphae enters the substratum and obtain the nourishment. Rhizoidal hyphae helps in anchorage. Rhizoidal hyphae behaves as root, whereas the stolon behaves as the stem in the angiospermy plants. In case of fungi, stolon is the structure which creeps horizontally on the substratum. The growth of the stolon is so fast so that it covers entire substratum. Rhizoidal hyphae, these are the finger-like structure which produced at regular intervals on the lower surface of the stolon. These hyphae enters the substratum and it obtains the nourishment. Not only that, it also helps in anchorage, that is fixation. Let us move towards the next part of the Rhizopus, that is the sporangiophore. These are the aerial, erect, unbranched hyphae produced when the mycelium enters asexual phase. Sporangiophores are produced in clusters opposite to rhizoidal hyphae. Each sporangiophore at its tips produces the sac-like structure called as the sporangia. Sporangia comprises sporangiospores in it. Let us move towards the reproduction. Rhizopus reproduces both by asexual method and sexual method. In this class, I am going to discuss with you about the asexual reproduction. In case of Rhizopus, asexual reproduction takes place by the formation of non-motile spores known as sporangiospores, which are produced in a sac-like structure called as the sporangium. Sporangium is developed at the apex of unbranched aerial hyphae called as the sporangiophore. Formation of sporangium and spores in it. During the sporangium formation, the tip of the sporangiophore starts to swell to form a spherical structure called as the rhibodium. This rhibodium enlarges to form a sac-like structure and now it is termed as sporangium. Within the sporangium, multinucleate with a dense cytoplasm, and these cytoplasm and the nucleus are moving towards the peripheral region of the sporangium. At This stage, the protoplast of the sporangium is differentiated into two regions, that is, outer peripheral sporiferous region with dense protoplast and central sterile region with vacuolate protoplast. These vacuoles forms a cleft between the sporiferous region and the central sterile region. The multinucleate protoplasm of the sporiferous region undergo cleavage, resulting number of sporangiospores. As the sporangiospores are black in color, so it is commonly called as black mold. The central sterile region forms the columella. Due to the production of sporangiospores in large quantity, a pressure is exerted on the sporangial wall which results in the release of the sporangiospores outside. These spores are carried away and fall on the suitable substratum like the bread and undergo germination, and the formation of new hyphae takes place. In case of the asexual reproduction, a stalk-like structure is produced called as the sporangiophore. This sporangiophore swells to form a structure called as the rhibodium, and this rhibodium enlarges to form a sac-like structure called as the sporangium. Within the sporangium, the spores are produced. In the beginning, these are multinucleated in condition. These nucleus are moving towards the peripheral region and forming the central sterile region. After the complete formation of the spores inside it, the sporangium bursts out and releases the spores outside. When it gets the suitable substratum, it starts to germinate and produces the new hyphae. This is the asexual reproduction in case of the Rhizopus. In the next class, I am going to discuss with you about the sexual reproduction in case of the Rhizopus. Thank you.